Okay, it is 11 a.m. Welcome to Indoor Ag Science Cafe. My name is Cherry Kubota. I'm the host for this webinar series hosted by three project teams funded by USDA NIFA. Thanks to USDA for supporting these activities, and then also thanks to all the project members who are supporting this also. Today's webinar is specifically funded by OptimIA Group, which is original group, started this Indoor Ag Science Cafe, optimizing indoor agriculture. This is for delivering scientific information to support indoor agriculture, including vertical farms, greenhouse, and also high tunnels. So therefore, I hope you enjoy this series if you are new to this. So just wanted to give you a quick update in terms of schedule. We had some schedule conflict, so we had to skip September Indoor Ag Science Cafe. But today is October Cafe, and then November, December. We do basically one of Tuesdays, and typically 11 a.m. Eastern time, Eastern US time. For those international, note that we will be switching to standard time. Daylight saving, I think, ends November the 3rd, and then starting the standard time from the November 12th cafe. So the November 12th, we will have Dr. Roberto Lopez, who is gonna introduce us the CEA Herb project. He's leading that project, multi-state project funded by USDA. And December, this is going to be hosted by ADVANCEA group. This is more like an engineering-oriented group working on greenhouse climate optimization. Dr. Mura Kachira from University of Arizona is going to talk about greenhouse technology, specifically what we call wavelength selective covering material, and then also PV integrated, so the photovoltaic, so the power generating greenhouse. So that's interesting technology. So Mura has been working on that years, so he's gonna share that information with us. And then more, cafe is coming up after January, so I will introduce that bit by bit in the future. All right, so, oops, the other way. So today's presenter is from Purdue University, Dr. Fatemeh Sheibani. If I pronounce your name right, you need to correct that. And then her title is Closed-canopy Lighting for Vertical Farms. Fatemeh has been working as an active member of this OptimIA group, and then it is so nice to have her as an independent scientist working in this very important technology for vertical farms. So with that, I'm going to stop sharing and then have Fatemeh share her screen. While she's doing that, just wanted to let you know the first part is recorded, and then we will stop recording as soon as her presentation ends. And then after that, it's an open discussion. You can ask anything without worrying about being recorded. And then also you can use the chat line if you feel more comfortable asking questions in the chat.
Hi, everyone, and thank you so much, Cherry, to have me today. It is truly my pleasure to be the speaker. I am one of the Indoor Ag Science Cafe fans from the beginning, and I've learned a lot from this platform over the past few years that you have this. And I'm happy to share my closed canopy lighting work that we have conducted in our lab a few years ago. Before jumping into the closed canopy lighting itself, I will provide some background information that probably you all know about it, but just to refresh your memory, As you know, vertical farming is growing rapidly in the global scale because it has many advantages, including higher productivity, significantly higher water use efficiency, minimized fossil fuel for the transportation because they are mostly located in urban areas, and we do have year-round fresh production. Based on some reports, the North America vertical farming market is expected to grow at the compound annual growth rate of 21% from 2024 to 2030. But on the other hand, there are concerns because this industry is generally considered to be expensive, high capital and operational expenses. Definitely the property in urban areas are expensive, which is mostly the location for the production side, and also the initial cost of LED installment plan. And along with labor, high electricity bill is another major operational cost. In this slide, I wanted to present some of the CEA census reports that we had access to based on 2019, 37% of vertical farms reported to be profitable. In 2020, that number increased to 47%. In 2021, 58% reported financial improvement, not specifically profitability, and 72% expected an improvement in the next year. And we all know that it was after pandemic. I couldn't find information of 2022, and 2023 was mostly focused on greenhouse production. I know that the 2024, this year, CEA census report was supposed to publish sometimes in October, and if it's published, I couldn't find that, but we are looking forward to that. So all these numbers that I presented here are very optimistic. This is one side of the story, and on the other hand, we see in the news or vertical farm daily news that some of the vertical farms ceased operation and filing for bankruptcy. So this profitability is still very fragile, and different aspects of vertical farming has been under investigation. In our lab specifically, we are mostly focusing on energy efficiency through sole source lighting. As we know, LEDs are the sole source lighting, and so far they are the best choice of lighting. Based on some studies, 25 to 50% of total operational cost is for electricity, and 52 to 80% of that is used for lighting system. The rest can be used for HVAC or utility lighting. These numbers might vary. The actual number might be even higher than these, but definitely we needed to have some references to present these numbers. And depending on the facility, this number might vary. But the bottom line is sole source lighting is one of the major burden on the operational cost of vertical farming. As we know, there has been tremendous effort and improvement on LED efficiency, and right now we do have LEDs that they are very highly energetically efficient, as well as they do have a good photon efficacy. Just to refresh your memory, electrical efficiency by definition is the ratio of optical power output divided by electrical power input. And the photon efficacy, which is a metric that we mostly use for LED, when we are using LEDs as a sole source of lighting for plant production, is micromoles of photon output per second and watts of input power. And as you know, a watt is joules per second, so the unit for that is micromole per joule. These two studies, one back in 2014 and the other one is in 2020, shows that how the photon efficacy of LEDs improved over time. If we look at these two graphs, and on the y-axis on 2014 it was above 2, and in 2020 it was close to 5. I couldn't find more updated information. Obviously, LED companies reporting their efficiency and efficacy in their catalogs, but I couldn't find a study that compiled all this information similar to these two studies and presented them. But the bottom line is LED photon efficacy are reaching their maximum theoretical values. So keeping that in mind, we need to think about that we are dealing with very efficient LED lighting system. The question is, do we use them wisely? In some cases, the answer is no, because we mostly forget about another term, which is canopy photon capture efficiency. The fraction of emitted photons reaching plant leaves and being utilized in the photosynthetic machinery. So we need to think that what percentage of those efficient photons, that tremendous effort has been done to reach that point, are using efficiently. That's why we think that rather than thinking about electrical efficiency and photon efficacy, we should think about unique physical properties of LEDs, that we kept two of those properties in mind when we were designing closed canopy lighting. Number one is LEDs are relatively cool at the emitting surface. They do produce heat, but that heat is not significant because they do have very effective heat sinking system. And since they are current driven, when they are dimmed down, when the amps are cut down, the energy expenditure declines significantly. So, and this is how the whole idea of closed canopy lighting shaped, and based on the definition, closed canopy lighting means when LED lighting system can be placed close to the foliar canopy without scorching leaves, and the minimum separation distance is determined by intensity and the spectral uniformity, which I will explain all of these later. But before talking about closed canopy lighting itself, we needed to learn more about the LED design. As you know, LEDs were first designed for human perception and for indoor application, and the same design is still being used for the plant production in indoor farms or vertical farms. An LED package, which is a single electrical unit, including one or two die with some thermal or some mechanical component, is a spherical source of light, which means that it radiates in 360 degrees. And after we mount LED on a flat surface, each LED is a hemispherical source of light, and it radiates in 180 degrees. Since LEDs are designed to be close to the ideal Lambertian or cosine weighted distribution, most of the illumination pattern, like 78% of the illumination, falls within 120 degree angle, or plus minus 60 degree. So as you can see in these polar coordinates, although there is an illumination beyond minus plus 60 degree, but this is not significant because most of the illumination happens within 120 degree angle. So there is a misconception that rain of photons falling from LED fixture and the separation distance between LED emitting surface and crop surface does not matter. But the reality is different. Depending on the LED package, The beam spread, this is the term that is mostly being advertised in the catalogs from the companies, is 120 to 140 degree. And when all LEDs are energized in a planar array, a new or unknown pattern of irradiation occurs that there is not enough working knowledge about that. So there are factors that affecting this beam spread. One of them is primary optic. Primary optic is a resin cap protecting LED dyes or a concave pit painted with reflective material such as titanium dioxide on mounting surface. When primary optic is used to protect LED dyes, the viewing angle or the beam spread is still 120. But when secondary optic is being used, which is a cylinder made with reflective material, and they install the secondary optic to help the uniformity of intensity and spectra. We know that LEDs are designed based on Lambertian. They are brighter in center, and this reflective material of the secondary optics helps to create a good combination and a good blend. But a secondary optic would cut down the beam angle to 60. So we have better uniformity, but we are losing efficiency. So as we have the truncated 60-degree beam spread, it means that we need to have more engines to have better uniformity throughout the fixture. So let's talk about closed canopy lighting itself. When we are talking about closed canopy lighting, we are suggesting that as the distance between crop surface and emitting surface of LEDs are decreasing, the chance of obliquely emitted photons escaping from the growth compartment is reduced. As we know, we do have 45 centimeter separation distance as the control because in vertical farm setting, the standard separation distance is between 40 to 50 centimeter. And we want to show that how these chance is getting less and less as we get closer and closer. On the top panel, you see the 120 beam spread, which is either naked dyes or the LED package with primary optics. And in the lower panel, you see the 60-degree beam spread using the secondary optics. And from left to right, you can see we have 4 different separation distances of 45, 35, 25, and 15 centimeter. As this distance is decreasing, the chance of photons escaping from the growth compartment is decreasing as well. And for the purpose of illustration, we only energized 3 LEDs, 1 in the center and 2 at the edge. So imagine that when all LEDs are energized, there is an overlap, and also these waste from the growth compartment is even higher. And we designed 2 scenarios of closed canopy lighting. Scenario 1 is energy efficient strategy, and scenario 2 is yield enhancement strategy. In the first scenario, we set LEDs at different separation distances, but we provided same intensity of light under each of them, which means that we cut down the energy expenditure. So we provided same intensity of light, same biomass was expected with significantly lower energy expended on LEDs. In the second scenario, which we did not dim down the LEDs because we know that in the specifically older generation of LEDs, dimmability is not an option. So as we reduce the separation distance, we kept the intensity at 1. I will explain later about these numbers, but basically we had higher intensity at closer separation distance which we expected to have higher yield, but we expended same amount of energy. I know it's a lot to digest on this slide, but I will explain more details in the coming slides. So, experimental layout and environmental inputs were constant for both scenarios. We conducted this experiment, the whole experiment, in a walk-in growth chamber. Relative humidity, temperature, and CO2 concentration were constant. We used red oak leaf lettuce rushi as the model plant, and to decide the cropping system, it was another side project because we wanted to develop our own lawn of baby greens. Baby green was our focus from the beginning. We knew that on day 15, after starting the seeds, is the stage that we can call them baby green based on the definition. And we wanted to have fewer photon loss for shorter time. But first, we needed to decide what material to use. And after doing some investigation on the different percentage of coco coir perlite, we ended up using 50% coco coir perlite by volume. And then we needed to decide what's the best seed density. We didn't want to broadcast seed to waste all those seeds, and we wanted to have the distance between plants somehow that the canopy closure would happen on day 15, which was the day that we defined them as baby greens. So we ended up using 84 seeds. It was a very robust and very low maintenance system, and it worked perfectly fine for this purpose and even for the other experiment that we conducted after this. So the only difference between these two scenarios was the LED adjustment. We had 4 BPSE LED lighting system inside the growth chamber. These LEDs were not designed for the purpose of closed canopy lighting, but because they were height adjustable, they worked perfectly fine, and we already have them in the lab, so we used them. And we considered 160 micromole per meter square per second as the total photon flux density combination of red, blue, and green. So we had 4 separation distance, 45 as the control, 35, 25, and 15 centimeter as the other tested separation distances. And as you notice, the total photon flux density was 160 under all of them. But it's interesting to look at the kilowatt hour of energy. This is the cumulative kilowatt hour of energy, which was 40 kilowatt hour for the 15 days cropping cycles to reach 160 micromole at 45 centimeter. This number was 31 kilowatt hour at 35 centimeter, 25 kilowatt hour at 25 centimeter, and 20 kilowatt hour at 15 centimeter. So the layout was like that. It was the same intensity of light, and we expected to see the same biomass production under each of these LEDs, which our statistical analysis proved that we see no significant differences in the fresh biomass produced at various separation distances. On these x-axis, you see different separation distances, and the y-axis is either fresh biomass and dry biomass. So as you notice, there is no significant difference in the fresh biomass production at various separation distances. Same for dry biomass, although there is a pattern there, but again, these dry biomass were not statistically significant from each other. So we are bringing energy into the picture. Our metric to define the productivity and the efficiency of this method was energy utilization efficiency. Gram fresh or dry biomass produced per kilowatt hour of energy. So As you see in the first graph, at 15 centimeter separation distance, we had the highest fresh biomass produced per kilowatt hour, and at 45 centimeter, the lowest fresh biomass produced per kilowatt hour. And the same trend is observed for the dry biomass. 15 centimeter has the highest energy utilization efficiency, followed by 25, 35, and 45 was the least. If I want to summarize that energy efficient strategy in one slide, this is the slide. So we had same PPFD. We expected to have same biomass, which we had, but we had less energy expenditure at closer distance. As you can see, there was no even visual difference between plants at 45, 35, 25, and 15 centimeter. And lower panel is the energy, 40 kilowatt hour, 31, 25, and 20. And when we pick 45 centimeter versus 15 centimeter, two extremes, 2.2 gram fresh mass per kilowatt hour produced under 45 centimeter and 4.4 gram fresh mass per kilowatt hour under 15 centimeter, which is a big significant difference between these two. So next is the yield enhancement strategy. What we did, we used the same setup, but the difference was we set all lights at 45 centimeter first and 160 micromole per meter square per second. And then before we start the experiment, I'm talking about the lighting map and this measurement. And then we decrease the distance to 35, 25, and 15 centimeter, and we use the spectroradiometer to measure the total photon flux density. So at 45 centimeter, the PPFD was 160. As we get to 35 centimeter, it jumped to 280, 350, and 480. But the interesting point is we expended the same amount of energy on lighting. The energy is 40 kilowatt hour for all the lighting system. And spectral composition and photoperiod were constant and similar to the other scenario. So we had higher intensity at closer separation distances, and we expected to have higher biomass, which our statistical analysis showed that we had the highest fresh biomass at 15 centimeter and lowest at 45 centimeter, and 25 and 35s are falling in between. The same pattern observed in the dry biomass. The highest dry biomass achieved under 15 centimeter, followed by 25, 35, and 45. And when we bring energy into the picture, it's getting even more interesting because the highest energy utilization efficiency, which is the function of gram fresh or dry biomass produced per kilowatt hour of energy, which is our standard metric, was highest for 15 centimeter and lowest for 45 centimeters. So these two graphs are very similar to the first scenario. And if I want to summarize this yield enhancement strategy in one slide, so we had higher PPFD at closer separation distance, higher biomass was expected and achieved, but same energy expenditure on LED lighting. So as you see, plants are visually different. We had higher biomass at the closest separation distance, energy was same. And when we calculate the fresh mass produced per kilowatt hour, comparing 45 to 15 centimeter, it was 2.1 gram versus 4.3 gram, which is a huge difference between these two. So at this point that we got our results with closed canopy lighting and we characterized closed canopy lighting, we thought that we can either even improve the closed canopy lighting. We knew that even at the closest separation distance of 15 centimeter, there is still photon loss beyond the cropping area. And we thought that if we add reflective curtain, some of those photons from the wide angle of 120 that are going to be lost, they can be recaptured and reflect back to the growth compartment. Although we know that the reflection law of physics does not exactly work in that case because we are not dealing with a mirror-like surfaces, but still those of the reflective material, and some of these photons can be recaptured and reflect back to the growth compartment. Because when we are thinking about the commercial scale, because of the intensity drop off with lateral beam spread from LEDs, we know that indoor growers extend LED arrays to the bench edges or beyond for the sake of crop uniformity and marketability. But there is a photon spillover not only from those edge engines, but also from the one near the center, far from the edge. On the other hand, we knew that when we are adding reflective curtain, it would restrict horizontal air movement, which is essential for thermal control, CO2 refreshment, and water vapor removal. So rather than picking 15 centimeter, which was the most efficient distance based on closed canopy lighting, we decided to go with the 25 centimeter separation distance, which was the second most efficient distance. And then we combined these 25 centimeter separation distance with different scenarios of closed canopy lighting. So in this first case, we set all LEDs at 25 centimeter separation distance. And as you remember, in the energy efficient strategy, in the first strategy, all of the lights were 160 micromole. So we set all of them at 160 micromole per meter square per second. And we surrounded two of these growth compartments with reflective curtain. And the front panel of these reflective curtain, as you see in this image, are fringed because we wanted to have some sort of air movement still. Then we conducted this experiment, the same cropping cycle of 15 days. And after 15 days, we noted that there was no significant differences between fresh biomass produced in those treatments that we used reflective curtain or without reflective curtain. Same for biomass. No significant differences was noted between those. We expected to see higher yield for those treatments that we surrounded the growth compartment with reflective curtain, but there was no significant differences. Although energy was not our focus, because we knew from the beginning that all of the LED system will use the same energy expenditure. So, but still we calculated energy utilization efficiency, and obviously since the biomass was not significantly different, the energy utilization efficiency was similar for these two as well. So as the next step, we combined the yield enhancement strategy. So as you remember, in the second strategy, which we call it yield enhancement, at 25 centimeter separation distance, the total photon flux density was 350. So we set all LEDs again at 25 centimeter, and we set light at 350 micromole per meter square per second. We surrounded two of these systems, as you see, with a reflective curtain and two without reflective curtain. And as you see here in these trays, there is a visual difference even between these two plants, that these two type of system that we had. And it was interesting that the combination of higher photon flux density and reflective curtain resulted in higher biomass. We had higher fresh biomass, significantly higher fresh biomass in those treatments that we surrounded the cropping system with reflective curtain. And same pattern was observed for dry biomass as well. Again, energy was not our focus, but obviously because the fresh or dry biomass was significantly higher, energy utilization efficiency was significantly higher for those treatments that we had reflective curtain. So as conclusions and future direction, with focus on LED lighting, CCL is an effective energy saving strategy for vertical farming, and yield enhancement strategy is a near-term benefit if dimmability is not an option, which we know in the older generation of LEDs, dimmability is not an option. CCL provides insight for the current application and future design of sole source LED lighting system. We learned that in the first place, LED fixture design is very, very important for the success of closed canopy lighting. We need to have a great uniformity of intensity and spectral composition. We do not want to have patches of colors or patches of brightness versus less bright area under LEDs as we get closer and closer. Obviously, we need to have an effective heat sinking system. And as I mentioned earlier, the primary and secondary optics would affect the shape or the beam shape, and ultimately it needed to be considered for the closed canopy lighting and for the future design of LEDs. And also those LED fixtures that the electrical component occupies a large area of emitting surface, those are not, I think in general, they are not good, and even they are worse for the closed canopy lighting, because as the distance between the lighting system and plants decreased, that shade band, which is the electrical component on the emitting surface of LEDs, are getting worse. And for example, if you have lawn of baby greens growing under that area, they would respond shade avoidance syndrome, and you have lower marketability and yield. Also, we believe that for the future of vertical farming system, height adjustable rack system should be an option. We might think about flexibility of CCL throughout cropping cycle, and it's another interesting area of research for the future direction. These are some questions that we frequently being asked when we talk about CCL. If the tip burn will be an issue when plants are grown beyond baby green stage. We did not have any issue with the baby green stage in terms of tip burn, and obviously Cherry is an expert on tip burn. We know that as we get closer, that the separation distance decreases, it's harder to remove the excess humidity, and as VPD decreases, the tip burn increases, and Cherry can comment on that better, obviously. So tip burn might be an issue if there is not a good air movement or air uniformity. So these are very important, both air movement, air uniformity, air direction to remove excess humidity, to fresh CO2 injection, and to maintain thermal control. And obviously all of these depends on the whole layout and bench like length and the whole layout of the vertical farming. And there should be an innovative engineering solution, for example, to integrating air movement with lighting. So these are some thoughts for engineers. We proved that closed canopy lighting can save a lot of energy, and there are obviously some things that needs to be addressed specifically by engineers. So integrating air movement with lighting is one of them. So right now I'm switching from closed canopy lighting to another area. So far, we have been talking about photon loss beyond cropping area. What about photon loss on the cropping area? For example, imagine that you do have indoor production of individual plants that you want to keep them for more mature stage. So you know from the beginning that you need more space later, and re-spacing is not an option. In that case, significant photon loss is happening during the early production, as you see in these images. When plants are very tiny, the photons are falling. Again, those very efficient photons are falling on the empty spaces between plants rather than being utilized by plants. That's why the whole idea of targeted lighting started in our lab a couple years ago, done by another graduate student. They were thinking how to minimize the photon loss on the cropping area, and if there is any engineering solution for LED lighting needed to be done in that regard. So in the targeted lighting study, rather than the full coverage, which is all LED engines are energized for the whole cropping cycle, they kept the engines on top of each plant energized. And this study showed that significant improvement in energy efficiency happened there. And based on that study, another study suggested that we should have the LED, a new LED engines or unit which has a central camera that can detect plant size, and based on that, different segments of LEDs are energized. For example, if plants are very small, the central segment is energized. As they are getting bigger, the outer segment is energized, and ultimately all of the segments are energized. So this would obviously add to the higher capital expenses. But another solution that we have been thinking about is we suggested that a single beam above each young plant can result in even higher energy efficiency when combined with a height adjustable lighting system. In the other word, the combination of targeted lighting, which was a single beam, with closed canopy lighting, scenario one, which we are dimming down the LEDs, can even result in the higher energy efficiency. These illustrations are done by our industry, by our engineering partner, Mike Bourget. And again, based on the Lambertian design of the LEDs, we know that they are brighter in the center, and even they are getting brighter as they are getting closer to the plants. So we think that we call it focused lighting, we are emphasizing on energy saving during early production, during the time that photosynthesizing tissues are very small and photons are falling on the space between plants. Then we do have a full coverage rather than being just focused on the plants. So we postulated that this focus lighting, which is the combination of a single beam on top of each plant and closed canopy lighting, would help those small seedlings to benefit from the bright center of LEDs. And as the plants are growing, the distance can be increased, and the wider beam angle could cover larger plants, and photons are being used very efficiently rather than being wasted. So this study is under review, and we will report more information in the upcoming platforms or events. With that, we would like to acknowledge USDA and OptimIA project. OptimIA was a great chance for us to investigate different approaches, not only closed canopy lighting, but also other studies we conducted. We also like to thank Sierra Space for the LED lighting system, RZ for the generous seed donation, and True Leaf Market. This article of closed canopy lighting was published last year. If you have any question or you would like to know more details about it, please feel free to take a look. So with that, I would open the floor for the questions. If you have any question right now, or if you would prefer to contact us later, Dr. Mitchell and myself would be happy. And Dr. Mitchell is here as well with us if he wants to make a comment or something, or you have a specific questions for him. So I would stop sharing and I would take questions if you have any.
Well, thank you so much. I really appreciate Fatemeh. It was very nice work. Congratulations.